Let me just hold on before you start recording. Just okay, that's fine. Okay, we're good.
All right, all set.
Good morning, everyone. Thank you so much for joining us today. My name is Miguel Garcia. I'm the Sustainable Ag Program Manager for the Napa County Resource Conservation District. And today we're going to be talking about fundamentals of soil health. What is soil health? How do you assess it? How do you enhance it? How do you maintain it? Our workshops are intentionally a little bit intended more for the vineyard industry, just because we're in Napa County and that's what I focus on. However, the principles that you're going to be learning today are going to apply to any other crop system that you're working on. So feel free to reach out to us if you have any questions. And today we're going to be having a few speakers from the Natural Resource Conservation Service, which is a branch of the USDA. And we are very fortunate that we share an office with them. So we collaborate a lot together in order to help you meet your objectives as a grower. Our first speaker today will be Evelyn Denson. Evelyn is a resource conservationist, and she will be talking about soil health. After that, we'll have Chip Burrow, who is our soil conservationist, and the last presentation will be by me. This is a 2-day workshop. So today we're gonna be, we're scheduled for 3 hours. We have about 2.5 hours of content, and then with conversations, we hope to fill in the rest. We ask you that any questions that you have, type them in on the chat box as the speaker is speaking, and I'll keep track of those questions. At the end of each presentation, we will have time for questions, and at the end of Chip and Evelyn's presentation, which will be about an hour, we'll have a break, and then we'll come back after that. So Evelyn, go ahead and share your screen, please. Evelyn, can you hear us?
Yep, I am. I'm in process of doing that.
All right, go ahead, sorry. Awesome. So while Evelyn sets up, please join us tomorrow. Tomorrow, we're going to be having a set of other speakers. We're going to be talking about soil erosion. We're going to be talking about how cheap grazing can benefit your soil health. We will be talking about biochar, compost, mulching, and we're going to be having a representative from the Carbon Cycle Institute talking about carbon farming. So tomorrow we have a very interesting agenda as well. Same time, 8 to 11. So please join us if you can. If not, that is also going to be recorded. All right, Evelyn, take it away.
Okay, so Chip and I both work here with the Natural Resources Conservation Service, as Miguel said, and we're here in Napa to assist any landowners and farmers who have questions about soil health. We're going to start out the presentation today with a quick background on soil and soil formation. So several processes are responsible for creating and forming the soils that we have, and it takes a lot of time for them to happen. There are 4 main processes. There's additions, and this could be, you know, if you had a windstorm and blowing dust particles, they'll settle somewhere else. They will, you know, become part of the soil in another location. You've got additions from weathering of the parent material that form our soils. You've also got losses when you have erosion and you lose soil from wind or water. You can have losses of nutrients in your soil from leaching. You have translocation of materials through your soil. That's the arrow that's sort of going down through the soil. That can be dissolved particles that are traveling with water through your soil. And you've got transformations when one soil particle can get converted through the physical or chemical processes into another. And there's several processes are responsible for these, or several factors cause these processes to happen. One of them is climate, and climate includes temperature and rainfall. Of course, the rainfall water is a real important factor for soil formation, as well as temperature in colder locations. The processes just don't happen as quickly as in warmer ones, though in cold locations, the freezing and thawing that you get can also be critical in forming the types of soils that you have. Organisms like bacteria, fungi, plants, and animals, well, those are all going to be active in forming your soils, and also organic matter and the reactions such as the transformations that happen in the soil. You've got relief, slope, and which is such as topography, that's going to affect your slope and your runoff, as well as your aspect and where the most sun is intercepting your soils. And again, that ties back into your temperature. Drainage is also important as far as forming the soils that you have. I think we're all aware that areas that don't drain as well, the soil may be different than areas that do drain. Your parent material, this is the bedrock that the soils are formed out of. That, through the processes of weathering, they will, go from bedrock into the soils. You can also have parent material that isn't bedrock, though. In an event where you have a large quantity of soil move, such as in a flood and a large erosion event, that's transported parent material. So it originated somewhere else and was transported to the location, but that's also considered parent material. Time is the final factor, and that it takes time for all of these processes to happen. Well, a lot of times we talk about young soils and old soils. So young soil, of course, less time has happened to affect all of these ongoing processes. But soil, it's even old soils, they're continually changing. It's never, the process is never done. And all these processes work together to create a huge diversity of soils throughout the world. We can influence soil formation by affecting climate. We can affect organisms. We can even affect relief by changing drainage. And with amendments, we can affect the chemical and physical processes that are responsible for the transformation. So there's a lot we can do to manage our soils and be sure that we're building healthy soils. When you dig down vertically into a soil, I'm sure that you're all aware that there's different profiles. The soil looks different at the surface than it does as you dig deeper into the soil. This is called your soil profile. And as soil forms and ages, it develops horizons or distinct layers, and you can see that when looking at a soil profile. An undisturbed soil has an O horizon on the surface. This is an organic layer that's made up of decomposing organic litter. This layer may not be present in a vineyard because we do tend to manage our soils more, but it would be a layer that you would definitely see in a forested ecosystem or a very undisturbed vineyard or orchard where you're not turning or disturbing the soil as much. You may see that if you have enough organic matter building up on the surface. Next down, you have the A horizon, and it's the top mineral horizon. What I mean by that is that there's some of the mineral soil that did originate from the bedrock. The O horizon is really just organic material. So the first mineral horizon, the A horizon, has mineral soil, and it has lots of biological activity still because you do have a lot more organic matter working its way into that A horizon. It's also the most influence from the climate because sunlight's going to be changing the temperature of that A horizon more than it will the soil deeper down. Also, water is going to be interacting with that top layer of the soil the most. It's enriched by the accumulation of organic matter, and it's the layer from which crop plants are going to obtain most of their water and nutrients. The B horizon is a zone of accumulation. It accumulates materials that leach into it from the upper layers. This can be organic matter, it can be various nutrients. Translocation can cause dissolved particles and nutrients to leach downward with the movement of water. And clay particles are also deposited into the B horizon. The formation of clay takes time, so in young soils, there may not be a B horizon, but as soil ages, this horizon does develop. The C horizon is below the B, and little biological activity happens here. It has a similar chemical composition to the parent material, and it's a zone of weathering as that parent material breaks down. The R horizon is the bedrock. We don't really show that here, but that's, below the C horizon. Really, the bedrock is, the base underneath all of the soil. So this is a pretty simplified soil profile worldwide with a variety of climates and vegetation. There are other horizons and also sub-horizons that exist, but here in Napa Valley, these will be the most important ones to be aware of. And on the right side of the image, you can see we have it separated somewhat from a zone of leaching from the top down to a zone of weathering at the bottom. And the leaching is, where all that organic material accumulates. Some of that is going to be dissolved in water and transfer down through translocation into the zone of accumulation in that B horizon. We also mentioned clay particles could do that. So we have the presentation today broken up into 3 sections: physical properties, biological, and chemical properties. I'll go ahead with the physical properties, and Chip's going to follow me with the biological and chemical soil properties. Soil texture is a description of the mineral particle size. Soil mineral particles that are active in supporting plant growth are divided up into 3 categories by size: sand, silt, and clay. Sand particles are the largest, silt are intermediate, and clay particles are the smallest. This table shows you the diameter size of these soil particles as well as their surface area. So very coarse sand can be up to about 2 millimeters in diameter, and you've got fine sand down to, say, a 0.1 to 0.2 millimeters in size. Silt particles are somewhere intermediate in size, and clay at the bottom you can see are very small. They're about 0.002 of a millimeter or smaller in diameter. And clay particles you do need an electron microscope to see, whereas I think we're all aware that sand particles you can just see with the naked eye. Per gram of soil, clay has thousands of times more surface area than silt. So that's the surface area of that particle. Because there's so many more particles, because they're so small, there's a lot greater surface area on the clay. And the arrows on the right-hand side, you can see that for a medium sand particle, you've got about 45 square centimeters per gram. You go down to silt, it's got 10 times as much, 454, and clay down here, 18,000 times more than the silt does. And so it's going to become important when we start talking about some of those chemical processes as well as soil water holding capacity, that there's such greater surface area on clay particles. This is the soil texture triangle. Soil texture is it's a description of the relative proportions of sand, silt, and clay, and that's what determines your soil texture. This triangle can be used to determine which of the 12 basic soil texture classes your soil belongs in, if you know the percent of sand, silt, and clay in your soil. And soil texture classes are things like clay, clay loam, loam, sandy loam, and they're divided up here. It takes a little bit of practice to figure out how to use this, but you've got 4 silt particles on one side of the triangle, sand at the bottom, and clay on the other side, and you can see a range going from 0 up to 100% for each of those particle types. If you knew your percent, you would follow the number in from each side to see where it meets in the middle, and that would tell you what your soil texture class is. The most common way to determine soil texture in the field is with the soil texture by feel method. Sandy particles feel gritty, silt feels smooth, and clay soil is sticky and can be molded. Of course, the majority of soils are a combination of all 3 particle sizes, but with a bit of practice, anyone can follow the soil texture by feel method to determine their soil texture. This process is best gone over in person in the field, so we're not going to go over it in detail now. Another way to determine what your percent sand, silt, and clay is with this web soil survey. It's a USDA resource for the public and can tell you the expected soil texture at your location. Bill is going to talk a little more about that web tool tomorrow. As an example of how to use the soil texture triangle, we can see a point in the middle here in loam. Let's say we knew we had 41% silt. Let's see, I think it says 46% sand and 13% clay. We would follow the lines in and see where it meets in the middle here, and that one would be classified as a loam. Sandy soils have, sandy soils can have more rapid water infiltration and really good aeration, but low water holding capacity. Clay soils retain more water and nutrients due to their high particle surface area, but they have much slower air and water movement through the soil. Loam is more intermediary and made up of somewhat equal percentages of sand, silt, and clay. And soil texture is one of the most important physical properties related to plant growth because it affects nutrient and water holding capacity. Soil components. Soil isn't just mineral matter or solid matter. In fact, solids make up just half of soil. And see on the left-hand side here of this pie chart, we've got mineral matter. And of that, a lot of it is mineral matter, actually, broken down parent material that's now turned into clay, silt, and sand. You also have a small amount that's organic matter. And the other half of your soil is really pore space that can be filled with either air or water. And ideally, it's a combination of both air and water for healthy plant growth. As soil wets up during rain, it has more water and less air, and as it dries down, air increases and water decreases. A small percent is organic matter, and that's mainly just in the top horizon of the soil. Soil structure is a description of the aggregates. Soil particles usually stick together into groups of particles known as an aggregate or a PED. Soil structure is the way in which aggregates are arranged. This chart shows a variety of different shapes of aggregates. Soil structure can develop as clay and organic matter increase because they act as binding agents. They stick individual particles together into aggregates. Sand itself, as we all know, it doesn't stick together very well, but if you add some clay or some organic matter to it, it starts sticking together more. You can see in this table that there's a variety of shapes. Single grains that have no binding agents are just known as single grains. They don't have really any clay or organic matter to stick together. As those binding agents increase, you can get a granular structure. If there's a compacted layer, you can get a platy structure where the lateral dimension is much greater than the vertical. That's the third one down. In a B horizon where the clay accumulates, you can get more of a blocky structure. In a C horizon, you can get PEDs that are termed massive, such as those down here at the bottom, with no definite structure or shape. Physical forces such as tilling can affect aggregation by breaking those PEDs apart. And climatic forces such as wetting and drying or freezing and thawing can also affect aggregate formation. Now, why do we care about soil aggregates and soil structure? Well, because it affects root growth, water infiltration, aeration, and biological activity, all important factors for healthy plant growth. We talked about all the pore space in soils, how really half of the soil should be pore space. Within soil aggregates, there's pores, and pores help move water, oxygen, and dissolve nutrients through the soil. They're also space for your roots to grow in. There are large pores called macropores, and small pores are micropores. Usually the micropores are the most important for water storage because water drains more quickly from the macropores. In a moist soil, the most ideal for plant growth, the macropores are filled with air, and the micropores are filled with water. A healthy soil should have a combination of both micro and macro pores. Mentioned a bit about soil compaction. What happens as soil gets compacted? That means that there's a lot less pore space. You're squishing all those particles together. Air and water can't move through the soil as easily, and roots require oxygen to respire, and they produce CO2 that needs to travel out through the soil. And microorganisms also are trying to live in there and respire as well. So they need that air and water movement as well. As these pores get squished and disappear, you lose that network of pathways for water and air to get into the soil and move. Overall, as you take away pore space, you reduce the ability of the soil to support healthy plant growth. Aggregates and compaction. As you further compact the soil, you destroy the aggregate structure as well. You're not just squishing the pores, but you're squishing those aggregates. This will significantly affect the movement of water through the soil. The surface of the soil will form a crust, as you see in the image on the right. In a heavily compacted soil, water will not infiltrate into the soil at all and fill those pores, but it'll just run off. As you can see in this picture, an aggregated soil has pore space continuing from the surface all the way down through the soil so water can infiltrate well. A compacted soil has a crust on the surface, and even if there are pores lower down, the water cannot access them, but it just runs off. One of the factors that helps bind aggregates together are actually fungal hyphae. In a healthy soil that's less disturbed, you have fungal hyphae throughout that topsoil. If you were to see them, which is most easy in more of a forested soil, it's almost a web of sort of a white web through the soil. And the fungal hyphae actually produce a substance called glomalin, which acts like a glue to bind the aggregates together. These soils have better water infiltration, and they're capable of holding on to water and nutrients better too. Fungal hyphae are common in undisturbed forest soils, but they're also in a vineyard soil managed without or with less soil disturbance. All this time spent building a healthy soil with good aggregate structure can be destroyed really quickly with tillage. Mechanically, it's going to destroy the aggregate structure, and at the bottom, plow depth, there will be a compacted layer. Not only that, but all the extra oxygen you add to the soil as you turn it over is not beneficial. That extra oxygen is going to cause any of the surviving biota microorganisms and whatnot that are in the soil to respire more quickly, leading to organic matter loss. Respiration is actually just those organisms, they're taking the carbon in the soil, and as they respire, they're turning that into CO2, a gas which will be lost to the atmosphere. And that's what we're showing, just all these little fires above the soil. That's the process of respiration, essentially the solid carbon from the organic matter getting turned into CO2. And this will also be a loss of nutrients and the fungal hyphae that helped hold the soil together into healthy soil aggregates that and thrived in an undisturbed soil, but they're going to be destroyed in the process of tillage. So as you can see, the 3 bullet points here are going to cause the biota to increase SOM oxidation, or that loss of organic matter. You're going to release stored nutrients, and you're going to destroy the fungal aggregate glues. Soil particles can act in different ways depending on the amount of soil or the amount of water in the soil. Water molecules like to stick together through a process called cohesion. Water molecules actually have a positive charge on one side and a negative on the other, and those positive and negatives help bond the water molecules together. And water is also held tightly to soil through soil particles through a process called adhesion. Adhesion is when 2 different molecules are attracted to each other. Cohesion when 2 like molecules are. So soil and water bonding is adhesion. Water and water, soil and soil is cohesion. This is especially true on clay particles because they have a negative charge, so they bond tightly to that positive side of a water molecule. Water molecules, I mentioned, have positive and negative charges, so that allows them to be both attracted to each other and the soil particles with a charge. In saturated soils, as the image on the left shows, all the pore space is filled with water, and all that water is actually pushing the soil particles apart from each other. In moist soils, there's thin films of water around all the soil particles because that water wants to stick to the soil. This is a process of adhesion and cohesion at work. The water is sticking to the soil because water molecules want to stick to each other. They're also sticking to each other. And that helps bind all the soil particles together. This is why moist soil holds together much better than either a dry soil or a really saturated wet soil. These forces work together to hold water in your soil against the force of gravity. Based on gravity alone, you'd think all the water would just want to drain away. But because these forces are actually pretty strong, a certain amount of water really wants to stick to the soil particles. And some of that is always going to be available to plant roots. Particle cohesion. So depending on your particle size, water and other things are going to stick to it at different rates. In a sandy soil, very little water is going to want to stick to those particles because they don't have a charge. And also we talked about, the effect of surface area. There's a lot more surface area on clay particles, which really allows them to hold tightly to a lot more water, as well as other things like nutrients. And because that charge, they have a much higher cation exchange capacity. Chip is going to talk a little bit more about that later as well. Let's see, next slide, soil water retention. When we talk about soil water in terms of plant availability, there's a few important terms. This looks like the same slide we looked at before, but just a little bit different. Field capacity and permanent wilting point are two important points as far as soil water retention. In saturated soils, a lot of water will drain away due to the force of gravity. And imagine picking up a wet sponge. Water drips out of it, but the sponge is still damp at the end. That point is called field capacity. That's how much water is left in your soil after gravity's pulled, a certain amount off. The water films on the soil particles in that center image are what we think of with field capacity. They're stuck together through cohesion and adhesion to the soil particles. And that's a situation where you do have a significant amount of plant available water, as well as the pore space filled with air, which the roots need for healthy growth as well. As the water that's held onto the soil particles gets used or evaporates, eventually the soil gets to a point where there's no more water available to plants. There can still be some water left on the sides of the soil particles, but it's bonded more tightly to the soil than the plants are able to pull it off. And this is what we termed permanent wilting point. At that point, there's no more water available to the plants. And that's what you get as you move from the center image to the right one. The adhesive forces binding the water to the soil particles are greater than the forces within the plant to draw that water up from the soil. So at permanent wilting point, there's still some water in the soil particles, but it's held too tightly for the plants to use it. And of course, in a dry soil, there's essentially no water left. Depending on the soil particle size and soil texture, that's the relative proportions of sand, silt, and clay particles, wilting point and field capacity are at different points along a scale of soil water content. So essentially, if you're going from a completely dry soil to a saturated soil with 100% of the pore space filled with water, the points that we call field capacity and permanent wilting point are at different points on this scale. Ideally, for healthy plant growth, you want them to be far apart. That means more of that water is available to plants. In sand, because there's very little adhesion between water and sand, between the water and the sand particles, there's a much smaller range. So that's the top bar here. You can see wilting point and field capacity are close together, and they're on the lower end of this range. So there's not that much plant available water if your soil is pure sand. A dense clay binds water so tightly in soil, and that's the middle here, that a significant amount of soil water content is not actually available to plants because the forces that bind it to the soil particles are too great for the plants to be able to pull that water off of the soil. A loam soil is ideal for plant available water. Permanent wilting point and field capacity are far apart, and there's the most plant available water. This graph is another way of showing that same point. We've got a line on the bottom for permanent wilting point and a line on the top for field capacity. In between is what we term that plant available water. Along the x-axis, we see a range in the soil particles from sand to clay. And you can see in the middle, or this is more your texture classes from a sand through loams to clay soil, that a loam in the middle, which has a combination of sand, silt, and clay, has the most plant available water. So when these lines are the farthest apart, there's the most available water. Now, we can't really change how much sand, silt, and clay we have in our soils, but one thing that we can affect is the amount of organic matter. We can build organic matter in our soils, and that's a good thing because organic matter is capable of holding on to plant available water too. It can actually hold 10 times its weight in water. That means for every 1% increase in organic matter in your soil can hold up to 25,000 gallons more available soil water per acre. Adding organic matter to soils requires a healthy soil biota and plant cover, and Chip is going to continue the presentation on soil biological properties.
Okay, good morning, everybody.
Sorry, Chip. Sorry. Before we move on,
okay.
I will address some questions for Evelyn. So if anybody else has any more questions for Evelyn before we move in with Chip, feel free to type them in. Somebody just wanted a clarification, Evelyn. I think the chat responded it, but somebody thought that the L in the CLORP the CLORPT equation, mean losses, but that equation, it's talking about the soil formation. So the CL stands for climate. We just want to clarify that. And then somebody's asking about fungal properties.
Sure, so the fungal properties, I think Chip will probably go into more detail with that in his biological properties presentation. But I touched on it a bit with the glomalin because it's really important in developing the soil aggregates, which is a more physical property. But that what you see is that it's really, with healthy soil, it's a combination of everything working together. So hopefully, I think Chip is going to cover fungal properties more and the influence of fungi in soil.
Okay, awesome. Thank you so much, Evelyn. So we're going to have Chip Burrow, our soil conservationist, speak next. So feel free to start typing your questions for him, or if you think of any other questions for Evelyn, we're going to have time at the end of Chip's presentation to discuss it more. All right, take it away, Chip. Thank you.
Okay, good morning again. And I'm going to start talking about biological properties in soils, which I find really exciting. Evelyn's going to be running the slides, so when it's time to move a slide, I will say slide. Okay, getting back to the soil components, that it's half empty space and half solids, that little bit of organic matter, do you remember that? That's the part that I'm going to talk about. It's really important in soil, of how soil functions. So next slide. We can talk about organic matter in soils, 4 different types. Do the green part of this pie chart, fresh residue, fallen leaves, freshly dead roots, all kinds of dead animals, things that aren't decomposed at all. And then the larger purple part, decomposing organic matter. This is like compost. It's actively being broken down by bacteria and funguses. Then there's that light yellow part, stabilized organic matter, which is called humus. And I'm going to question that. We'll talk about that later. But this is the carbon in soil that's stable over long time periods. And it's important as it helps with the structure of soil, and it stores a lot of nutrients. And then the little orange part, the living organisms, which they make all this happen, and they're important too. Next slide. So soil biota, this is all the live things in soil. And the soil's a thin skin on the surface of the Earth's biosphere, but it's an important component of the biosphere. And there's a size range from bacteria, tiny, small things in the bottom, the number 10, up through, which are invisible, and up through more medium things that are tiny, tiny animals, and then up toward the top are things like earwigs and worms and such that we can definitely see. So they're all part of the soil biota, and there's this huge difference in size, and the worms and earwigs we can see, but most of these other things are pretty small and invisible. We normally don't see them, but they are there. Next slide. This slide makes the point that these small invisible things, the bottom is square meter, they're huge numbers of bacteria and actinomycetes, protozoas and such. And as the animals get bigger, there are fewer of them. The larger animals are feeding off the smaller animals, or the bacteria and fungi. And this just goes up. It just makes the point that, in a square meter, you only have so many snails and slugs, and you only have one bird eating those snails and slugs. So next slide. This goes in a healthy soil. it's one with the 50% empty space and good organic matter in it. There are huge numbers of these organisms, and the 4 to 8 billion bacteria, 20 million of these actinomycetes, these kind of a thread bacteria, 1 million fungi, 200,000 algae, and 2,500 feet of the fungal hyphae. Those are the fungal threads that are sometimes visible as that kind of white stringy stuff, but mostly invisible. That's 1/2 mile of fungal threads in a teaspoon of soil. So there's a lot going on there. Most of these soil biota are saprophytic. They're feeding off dead organic matter, but they're very critical to nutrient storage and exchange and extracting nutrients. And just a factoid, soil contains 1/4 of the Earth's biodiversity. Most soil organisms haven't even been yet identified. So the soil food web, all this soil activity is fed by the sun, by photosynthesis. So sun in the upper left is growing plants, and through photosynthesis, they capture carbon out of the atmosphere from CO2, and they put their organic matter into the soil. All these bacteria and protozoa and fungi and all these things need carbon to eat. Plants produce carbon. We, like all these other microbiota, need to eat carbon to stay alive. So it's a different metabolism. So the plants are putting carbon into the soil, and the bacteria are using it. And when they're done with it, it releases as carbon dioxide. Let's see. And this slide also mentions that, you know, this energy that the plants are putting in is transferred upward to the larger animals. Again, same point. Next slide. And just again, that the energy for all this is coming from the plants. So having a growing plant on the soil is benefiting the soil. If you have bare soil, the kind of agriculture that just leaves an area fallow without any plants in the hope that it'll store some more rainwater, it's not improving the biology of the soil. Next. And sort of remaking the point that the plants are adding organic matter to the soil for their own benefits. And part of it is through roots and fallen leaves and dead leaves and such, but parts of the plants also put sugar into the soil through their roots, and we'll talk about that more. But you can see all these relationships of organic matter going into the soil, and that the result is a benefit to the plants. So plants benefit from all the work they do putting carbon into the soil. Next. So the rhizosphere, rhizo is the root, and the rhizosphere is that portion of the soil that's very near the root. And there are a lot of interactions between the soil and the roots. And you see the little bacteria and fungi there. There's the root cap and root border cells. As a root pushes through the soil, it kind of sloughs off skin cells of the root as it moves, and that's another way of putting organic matter into the soil. So but the plant influences the soil around its root by exuding sugars, amino acids, polysaccharides, and these border cells. So it's making the soil around the root much more attractive to all these bacteria and fungi. And the bacteria and fungi are bringing water toward the root, breaking down nutrients off the mineral soil and bringing them toward the root. then which is an amazing relationship. But in addition, on the right, these arbuscular mycorrhizae. There are funguses that grow into the root from the soil and into the cells in the root. And next slide. These go right into the root cells. So the root isn't gathering the water and nutrients in this case. The fungus, the mycorrhizae, are bringing it in from the soil and putting it inside the cells in the plant root. And like the little guys up top, the plant is feeding sugar to the soil, and the soil is feeding nutrients back to the plant. And it's the fungus in the soil that's doing that work. So it's this great relationship. It's kind of a symbiotic thing. Lichens that have algae and fungus that benefit each other. This is fungus and plants that are also benefiting each other. And this mutualistic relationship is present in more than 80% of the modern terrestrial plant families. And if you recall, mushroomers know that a certain kind of mushroom grows under a certain kind of tree, like a fir tree in the forest. That's because that fungus of that type of mushroom has a relationship with that kind of tree, so they know the difference. And okay, next. So stable organic matter, we all want higher organic matter in our soil because it helps with aggregation, it helps with nutrients, it helps with water storage. The old view was that organic matter in the soil, the leaves and roots and such, would just gradually break down into smaller and smaller pieces, and that they would end up being humic acid, humic substances, and such. And it turns out that that was not factual. It was what was easily measured in soil. So we talk about humus in soil, but it's not necessarily just tiny or broken down pieces of organic matter. Next slide. So the emerging view, and this is from UC Davis, is that the bacteria and fungi are breaking down, starting in the lower left with the plant material coming from the tree. They grow by feeding on the organic matter, and their population increases if things go well. And then when they use up the organic matter, they starve and they die and decay. And it's their cell walls that make up the stable organic matter. And it gets, there's more processes that make it last longer. But the idea that the good, stable organic matter in soil is not coming directly from plants, but it's coming through these bacteria and fungi. And it's their body parts that make the stable organic matter. Okay, next. Okay, chemical properties. I'm going to be very brief on this, but we'll go through some of the basics. Next. Soil pH, which is the relative acidity on the left side with those lower numbers, 4, 4.5, or a very high pH or alkalinity on the right, 9.5 or 10. This is a standard chart that most of the nutrients needed, and the N is nitrogen, P phosphorus, K potassium, etc., are most available when the pH is around neutral, which is 7. So maybe 5.5 to 7.5, 6.5 to 7 is ideal soil pH. And soils can naturally develop higher or lower pHs than that. Organic matter in the soil tends to neutralize the pH, and a lot of organic matter tends to stabilize the pH in soil, which is helpful to gardeners. Next. And cation exchange capacity. This, a clay particle has negative charges on its surface. And you remember from what Evelyn said, the surface of clay particles in soil is tremendous because they're such tiny particles and they're flat sheets. They're invisible. We can't see them unless they're in bulk. And because it has negative charges, it holds on its surface positive charged ions. And there's some of them shown here on the far left. It's ammonium going down to the right, calcium, potassium. It's hydrogen, it also holds water, magnesium, sodium, salt, which we don't want that much in our soil. So these are great storage vehicles in soil. The soil water solution can only hold so many nutrients. It'll leach out otherwise, but they're attracted to the surface of the clay particles, and they're attracted to the surface of these tiny, stable organic matter particles, humus. This is an old slide from old school. And I think Evelyn made the point. So in our soil, we want to store as many nutrients as we can. We don't want them leaching out. So we want them to be available for plants. And typically in our soil, we cannot increase the amount of clay available, but we can increase the amount of organic matter. So that's an important reason to make sure that there's lots of organic matter in soils. Next slide. So the nutrient cycling, and this talks about nitrogen, but would apply to others, is if you just threw some chemical nitrogen fertilizer on soil, the plant is not able to immediately take up all of that or much of it. And this says less than 50%. Ideally, the nitrogen is taken up by the microorganisms in the soil and on the organic matter and stored on the clay particles. And then the plant can get at them gradually as the plant needs them. So the point there is the underline that whatever the nutrients, whatever their initial form coming into soil, they're usually being cycled through microbes before they get into the plant. So again, why microbes are important in our soil. Next. Just want to distinguish between fertilizers are the chemical nutrients, and they directly affect plant growth and add them to the soil, versus an amendment, which is typically mostly carbon, organic matter, but mulch, compost, manure, and they more indirectly affect plant growth because they increase the soil biota, they increase the water holding capacity, they increase the nutrient holding capacity, increase the aggregation, pore space, all these good things, and they add nutrients for slow release, which is better for the plants. And these are all big benefits. So there are nutrients coming in with amendments, but they're cycled through the soil biota first, and then they are much better available for the plants. Next. Okay, soil health, which is kind of the subject here, is a term the NRCS uses to summarize the characteristics of a healthy soil. And the point is that a healthy soil fosters healthy plant growth. Next. And the NRCS has 4 principles of soil health that are useful. The upper left maximize living roots on the soil, and that would mean plants. So always have a plant growing on the soil, and they have crop rotation, cover crops, kind of things. So then the lower left is maximizing biodiversity. And again, there are different techniques to do that. The more different kind of plants and animals that are interacting with the soil, the healthier that soil is. And that's macro animals like a cow, and also the microbiota. So let's go to the lower right. Maximizing soil cover is having plants or something on the surface. So it could be mulching, it could be leaves, anything on the surface. Protects the soil from direct sunlight. It keeps moisture in the soil. It keeps rainfall from causing erosion. So a lot of benefits there. And then the upper right, the last one, is minimizing soil disturbance. And it's kind of tillage or driving over soil with heavy tractors when it's wet. Disadvantage to that. Then next slide. So in this soil health workshop, which of these 4 principles can be used by Vineyards? And I've tried to highlight a few that I think might be applicable with little red arrows. And this is just for your consideration. I'm sure these are going to be talked about later, but these are some, techniques and practices that I've seen Vineyards use. And I think they'll, Miguel and others will probably be talking about some of these later. The grapes are, starting again at the upper left, grapes are a perennial crop. And most vineyards do use a cover crop, at least during the winter, to help protect the soil from erosion. Integrated pest management, starting lower left. Pollinator plantings, people put in hedgerows. They put legumes in the cover crop mix. We're talking about livestock use in vineyards. Let's go to the lower right. Vineyards use mulching sometimes. A lot of vineyards are trying to reduce their tillage, try to leave a little residue on the surface, certainly cover crops again. And then the upper right, minimize disturbance. Some vineyards are going to no-till. Everyone tries to stay off the soil when it's too wet. And it's a money saver. If you don't have to drive a tractor down a row, you don't have to pay the cost of that. So next. So this is just our contact info. If anyone is interested in contacting us, NRCS does technical advice on natural resource matters. It can also help farmers sometimes financially with cost share on some conservation practices. But I hope this gets you ready to hear more about what you can do to evaluate the soil health and improve the soil health in your agriculture. So, Miguel.
Thank you so much, Chip. So we're going to go ahead and play a couple of videos for you, showing you a little bit of these concepts that Chip has been talking about. Ideally, we will do this workshop maybe in a classroom setting and then take you guys into the field so that we can do this live. Unfortunately, given the current conditions, we cannot do that. So we decided to go out there, Chip, Evelyn, and myself, and we just recorded these videos for you. So I hope that you guys enjoy them. Okay, Ashley, are you there?
Yes, I am here and I'm ready to go.
Okay, go ahead and play the first one.
Can you see that one okay?
Yes.
So this test is called the slake test, and it measures the stability of aggregates in the soil. And we have these aggregates we've taken from the Gamble Ranch near Yountville, where they are conducting trials, tilling an area, and then 20 feet away is another block that has not been tilled, and I think it's over 10 years. So we took samples from there. The jars are marked. This head is tilled, that one is not. I don't see too much difference between the two. There's some pores in there. There appear to be a little more pores there. This one kind of got dustier and rounded, this one not so much. But we'll see how stable they are. When dipped in water, the water rushes into the pores. And if the fungal glues and such are holding it together, it'll stay in place. If those don't exist, once all the pores fill with water, the particles just fall apart. So let's see what the difference is. Ready? A little difficult to get in. Okay. This is somewhat representative of what happens when the soil gets a lot of rain on it in the winter. The tilled soil here in this example kind of comes apart and would form a solid sheet over the top, which would probably cause more runoff to happen. This one is more stable. There are pores in there, but it's not coming apart, so the pores will stay intact during heavy rainfall, and water can run into the soil and be stored in the soil rather than run off the surface. And what we could do in recording is, stop now and start again in another 3 minutes or 5 minutes and see how it's progressed. Also, there's not loose sediment in this one. In this one, a lot of soil particles are broken down to their individual particles, and that's why it's cloudy. The little crumbs that are broken loose on this are still stable in themselves, and they're just dropping to the bottom.
Awesome. Let's go ahead and play the next one, Ashley, please. And Chip, we're gonna need you to narrate on this one.
Okay, this test is called a rainfall simulator. And again, we use a kind of tilled and a no-tilled. And these samples are from the RCD's Wichita Creek Vineyard. And that's a tilled sample. The one there is untilled, which you can see also has more vegetation at the surface. And the idea is water is going to be put in the top trays, and it's got little holes, so it drips through onto the soil samples very quickly. And water that infiltrates is going to go into the rear plastic trays under the screen, and the water that runs off the surface is going to come out into these trays in the front. And like the slake test, we're seeing which causes cloudiness in the water and which keeps the water clear. You see what, I guess showing that, you can see the water is cloudier here. The soil at the Wichita Creek Vineyard is a clayier soil than at the Gamble Ranch, so there's a little difference in the way it goes. And more on the right, the untilled, more of the water went through the soil, absorbed into the soil, and less ran off the surface. And the stuff that ran off the surface was clear. So this is another way of showing the benefits of the good, stable aggregate that comes from less tilling and the benefits that you get from that. Both these demonstrations, the slike test and this rainfall simulator, we have the equipment to do this, and we'd be happy to do this for groups who might be interested in seeing this, both of these. So, good.
Awesome. Thank you so much, Chip. So we do have a couple questions for you here, and I'm just going to go ahead and read them. Carol is asking, organic matter throughout the upper levels of the soil as well as deeper in the soil?
I'm not sure I heard that.
Is the stable organic matter throughout the upper levels of the soil as well as deeper in the soil? I think they're asking if you find stable organic matter both on top and as you go down in the soil profile.
It's most of the organic matter is in that upper A horizon, if you recall Evelyn's slide, which is the zone of leaching. And then there's some organic matter in the B horizon also. Typically there's more in the A horizon, but of the organic matter in the B horizon, it's going to be a greater amount of the stabilized organic matter. And up in the A horizon, it's going to be more of that in-process decomposing organic matter. And even that O horizon that we don't have that much that around here, on the surface, is mostly undecomposed organic matter.
One more question. We have Marty asking about nitrogen fixing bacteria. He would like you to talk about how nitrogen fertilizer can impact the plant fungi relationship and lower soil organics due to scavenging for carbon by soil microbes. So I think he's referring to the carbon nitrogen ratio here.
Yeah. Adding a lot of chemical nitrogen fertilizer harms the soil microbiota. So it reduces them, the amount of, amount of them and their function, which later is kind of harmful to the plants. So I'm forgetting the rest of the question here.
I think you addressed it fine. So I think the question is, if you have, not only if it's an artificial nitrogen fertilizer, but if you have an excess of nitrogen in relationship to carbon, how that can affect your nitrogen and your carbon distribution. So if your carbon to nitrogen ratio is out of whack, what would happen?
If there's all carbon and no nitrogen, and this is kind of like the recipe for compost. If there's not enough nitrogen for the bacteria and fungi to live, there'll be fewer of them, their populations will be lower, and the organic matter will more slowly decompose over time. And the plants may, during that time period, the plants may be short on nitrogen because the bacteria in the soil are trying to grab it all to use themselves. If there's the right balance between the two, everybody's happy. The plant's happy, the bacteria are happy, and they can decompose the organic matter. If there's too much nitrogen and not enough carbon, the nitrogen stays in solution. It's not taken up by plant roots or microorganisms, and it leaches out of the soil, and it'll leach into groundwater. So it's a waste of nitrogen in effect. I remember the legumes was part of the question, is these bacteria that colonize legume plants, and they convert atmospheric nitrogen, which is not usable by plants, into other forms that are usable, but it's done very slowly and gradually, and it helps the plants, and it helps the microbiota nearby, and helps the soil.
That's great. And I think that it's important to point out as well that this ratio between carbon and nitrogen is not a 1-to-1. Usually, and please correct me if I'm wrong, Chip, but we're looking at 11-to-1. So a ratio of 11:1 carbon to nitrogen is usually what we look into to create that balance. When you go below that or higher than that, that's when you start seeing some issues with the carbon and the nitrogen uptake, correct?
Yeah, that's 10:1, 20:1, somewhere in there. If there's less nitrogen than that, it's scavenged, it's not available. If there's more nitrogen than that, it's wasted. So those are the ideals.
Awesome. Okay, well, Morty, I think we covered your question, but if we didn't, please let us know and then we can address it again. Thank you so much, Chip and Evelyn. We're gonna go ahead and say goodbye to Chip and Evelyn, but I think they're gonna stick around. We share the office, so if you guys were one-stop shop for you, if you have any questions for either of us, you guys are more than welcome. Our office is downtown Napa in Jefferson Street, or you can email us if you have any questions. So I'm gonna go ahead and do is, let's go ahead and send everybody into a small break. So I'm gonna give all of you guys 10 minutes just so that you guys can stretch. You need to go to the restroom, drink some water. And then when we come back, I will be talking about soil health. Just a reminder again, this is the first day of a 2-day workshop. We're gonna be coming back tomorrow with other speakers talking about soil erosion, biochar, compost, mulching, carbon sequestration, among other things. So please, if you get a chance, join us again. So I will go ahead and say that we're gonna come back at 9:25. So I will see you guys soon. I'm gonna go ahead and share my screen so that we can start my portion of the presentation. Okay, well, for those that join us a little bit later, my name is Miguel Garcia. I am the Sustainable Ag Program Manager for the Napa County Resource Conservation District. I added here my contact information. Feel free to reach out to me if you have any questions. So Evelyn and Chip talked to us about general soil properties and how those properties are used to assess and determine soil health. So what I'm gonna do right now is I'm gonna take that information and I'm gonna expand a little bit more on how do we assess soil health, how do we maintain it, and first of all, how do we define soil health. But before that, let me share with you guys something very interesting that we are doing. Some of you may know, but some of you may not know. The Napa County Resource Conservation District partnered up with the RCDs from Mendocino, Sonoma, and also the USDA NRCS offices to establish what we call the North Coast Soil Health Hub. And the purpose of this is to develop a network of farmers, industry, representatives, scientists, and agricultural professionals to work together to improve soil health on vineyards. Eventually, we're going to expand this work into other crop systems, but for right now, we're focusing on the vineyards. And our goal is to increase the adoption of regionally appropriate practices to help sustain the North Coast as a world-class viticulture region. There's no better way to achieve this goal than through improving soil health, which will help farmers increase farm resiliency, stay ahead of their regulatory curves, save money, and enhance marketability. The hub coordinates opportunities for hands-on learning and farmer-to-farmer communication and mentoring. I really encourage you to look at least at the website, sign up for the newsletter. We're trying to create this centralized location where all of this information for vineyards in the North Coast can be put together. We also established a network of 9 demonstration trials on vineyards across the region to optimize soil health practices for our conditions. The practices that emphasized in these trials are very fundamental in sustainable farming systems and include cover cropping, conservation tillage, compost applications. Besides this, what we're doing is we're collecting hundreds of soil samples. This project has been going on for several years now. And over this time, we are close to collecting 500 soil samples across the North Coast. This is from about 100 different vineyards. And our objective is to put all of this together into a database. where we can eventually share that with you. We're getting very close to that objective. And in the next coming years, we're going to be able to share with you this database of what soil health looks like for the North Coast region. And this is very important because right now, if you pay someone or you collect soil samples and send them to a lab, or you get a soil health assessment, you'll get some information. However, you're not going to know really how to go from there, because a lot of the recommendations that you may get are very generalized. Particularly right now, we might be comparing it to a study that was done by Cornell University in New York, and their soils are very different than our soils. So what we want to do with this project is essentially calibrate everything to the North Coast. What a healthy soil in a vineyard in the North Coast region looks like. And at the same time, we are trying to explore how different management practices affect these soil health properties. So what my presentation is going to be about today is looking at, first of all, how do we define soil health, but also what are we doing in order to understand better what different management practices are doing to our soil. So this is essentially a list of the partners. We have Sonoma, Goldrich RCD, both of them in Sonoma County. We have Mendocino. Definitely an important partner is the NRCS, which is a branch of the USDA. And we are partnering up with Oregon State University, which they are the ones doing the actual soil analysis for us. We are looking at most of the North Coast region. In the near future, we're probably going to incorporate Lake County and also Marin County. And when we do that, we'll expand a little bit from just vineyards into looking at rangeland and other crop systems. So a really quick recap of what Chip was mentioning about soil health. In general terms, we define a healthy soil as a soil that can function, a soil that can do its job properly. And in the case of an agricultural soil, we're looking at nutrient cycling, proper physical support for the vines, store and filter water, sustain life, microbiological life, and resist erosion. And when we're looking at soil health, we like to break it down into three different properties, which would be the physical, the chemical, and the biological. And with these three properties, we can essentially describe an entire soil health parameter for any given soil. So in the physical, we have aggregate and structure, which Chip demonstrated with the video that we showed you about till versus non-till. When you put it in water, how well those aggregates can sustain water and wind erosion. Surface sealing, compaction, porosity, water movement, and availability. In the chemical properties, we look at pH, soluble salts, nutrients, nutrient holding capacity, nutrient availability. And in the biological, we're going to look at macrofauna, microorganisms, roots, biological activity, and organic matter. So soil health defined in this way is a soil that can do its job, essentially, and we break it down into physical, chemical, and biological parameters. And when we do this, we based it on the USDA recommendations as far as how can you improve and maintain soil health. And we look at the 4 parameters that Chip mentioned. So minimize disturbance, maximize soil cover, maximize biodiversity, and maximize presence of living roots. So everything that we're going to be talking about, if you focus on these 4 parameters, you can improve whatever you are seeing that your soil is not being able to do. So if you think your soil health, it's not where it should be or where you would like it to be, these are the 4 parameters that I would recommend you look at in order to improve the biological, the physical, and the chemical aspects of your soil. So for the project that we've been doing, we had to define soil health a little bit more precisely. So the way that we decided to define it is a combination of different parameters. And these are test analysis that you can conduct on soils. And this is essentially the laundry list that we go through in order to determine if a soil is healthy or not. We look at water aggregate stability, which was represented by the video that Chip just showed, pH, nutrient storage and mobility. And we are focusing mostly on the macronutrients, so nitrogen, phosphorus, potassium, calcium, and magnesium. We focus on cation exchange capacity, which Chip demonstrated. Total carbon. And the reason we focus on carbon is because carbon, it's right at the center of all physical, biological, and chemical properties. If I can look at all of these tests, but if I only look at carbon, that gives me a pretty good indication of how the other parameters are going to affect it. If you don't have enough carbon in your soil, your water aggregate stability is going to go down just because there's not enough organic glues to keep your structure together. Also, it helps on the storage of water. It helps on the cation exchange capacity. So if I would have to pick one parameter to demonstrate soil health, that would be total carbon. But we're focusing on the other ones as well. The other one would be active organic carbon, potentially mineralizable nitrogen, bulk density, water infiltration, compaction, and respiration. So we go through this checklist and we look at how your soil performs on all of these parameters, and that way we give you an indication of how your soil is performing. Now, for the study that we're performing, where we are collecting soil samples throughout the region, we're not only interested on how these different parameters show up in the soil throughout the region, but we're interested to see how different management practices will affect these important parameters. And most importantly, we're looking at tillage, cover cropping, and herbicide applications under the vine. So what I'm gonna do next is I'm gonna walk you through what we have so far, and be mindful that these are preliminary results. We're still collecting more samples. We're still analyzing the data a little bit better, but this will give you a pretty good indication of what we're doing and what we're finding so far. So this would be the representation of 417 soil samples that we have collected from about 100 vineyards so far throughout the North Coast. What I'm going to show you is the averages of all of these soil samples. So the first parameter that we're going to be looking at is pH. And pH is very important because it influences nutrient availability, the soil biology, and the plant health. pH is one of those parameters that, in my opinion, get a little bit overlooked. Sometimes a lot of people like collecting perio samples from vineyards and making decisions as far as what kind of nutrients they're going to be applying. And in turn, sometimes they neglect collecting soil samples. A lot of people will collect soil samples at the beginning when they're developing a vineyard, and then they forget about it and then they never do it again. But in some cases, pH may be changing quite drastically from one season to the other or over time. So if it's been more than 3 to 5 years since you've collected soil samples and sent them to a lab, I highly recommend that you do this. One reason for that is because pH will affect the availability of nutrients. There's certain nutrients that the availability will be affected depending on the pH. If the pH is too high or too low, certain nutrients will not be available. And one example of that will be phosphate. If the pH falls below around 6.5, your phosphate begins to be locked in the soil. So it doesn't matter how much more phosphate you add to your soil. If your pH is not adequate, meaning above 6.5, then none of that phosphate will be available to the plant. So when you go and take your perio samples, you might see phosphate deficiencies, and the first response to that would be to add phosphate to the soil or apply a foliar or whatever method you use, when in reality, all you have to do is to fix your pH. So it is a very important parameter. And in here, this is essentially what the rest of the presentation is going to look like. I'm going to give you a parameter, and then I'm going to describe it, and then I'm going to show you the different experiments or samples that we are collecting. So here we have no-till, we have till, and then this is for the metals, and then the other three are for under the vine. So we have cultivation, herbicide, and just mowing. And in this case for pH, there's not a lot of differences, really. I mean, going from 6.8 to 7.3, that is not a lot of change. So the conclusion of this is that none of these management practices really affect pH. pH is one of those parameters that will change over time, just naturally given your soil properties. The next parameter that we're looking at is water aggregate stability. This is the representation of the test that Chip shows on the video. And this influences water infiltration, oxygen movement into the soil, but also changes relatively rapidly. So this is something that we definitely need to keep an eye on from season to season or every so often. And this is affected by organic matter. If you don't have enough organic matter, your water aggregate stability is going to be down. If you have sufficient organic matter, then your water aggregate stability is going to go up. So in this case, we are seeing that tilling, and this is in percentage. So this is the percentage of the aggregates that stay intact upon addition of heavy rain or wind erosion. So we're seeing here that tilling gives you a much less aggregate stability than no-till. And then if we compare the spray versus the no spray, we don't have data for mowing in this case, but we can see that if you till, you're going to have a reduction in your water aggregate stability. And that makes sense because every time we're tilling, we're disrupting the structure of these aggregates and we are releasing carbon from the soil into the atmosphere. So that is reducing the amount of organic matter in the soil. And in the case of the undervine, we saw on these that spraying herbicides, we didn't distinguish between which kind of herbicide. So this could be even an organic herbicide, but essentially removing all the cover crop under the vine will significantly affect your water aggregate stability compared to no spray. So in this case, a no-till system with no herbicide application under the vine will give you the best aggregate stability for your soil. And in return, this will allow you to have less impact of erosion, more water infiltration, water retention, and oxygen movement in the soil. A very important parameter. The next one we look at is the nutrients, and particularly we focus on NPK, calcium, and magnesium. And this is important because this is what's feeding the plant. So if you don't have enough, not only enough nutrients present, but enough nutrient holding capacity, and if your soils cannot distribute nutrients efficiently, then you're going to have some issues. So if we start with nitrogen first, this is in percentage, not a lot of nitrogen present in vineyard soils, not a lot of nitrogen needs in vines, but we can see that there's some differences between the no-till and the till. And in this case, we're seeing that no-till tends to have higher nitrogen concentrations than the till. And then we have cultivation, herbicide, and mow. So we can see that mowing just under the vine, leaving those living roots that Chip was mentioning that are so important, it's allowing for more nitrogen. And this can be many different explanations. It could be just the fact that your microbes are happier, that not tilling and not using herbicides will also increase your biological activity, and in return, you will have more nitrogen fixation. My objective with this presentation is not to tell you what to do as far as your management. It's just to show you what we are observing. This is the average of over 400 soil samples collected in Napa, Mendocino, and Sonoma County, so that you can use this information to better assist on your decision making. When we look at phosphorus also, we are seeing that for some reason, there's a huge increase in just mowing versus herbicide cultivation. We're seeing that no-till actually has less phosphate available than till. And I'm not going to attempt to explain why that is right now. Like I said, these are preliminary results. Eventually, we'll be able to explain this in a lot more detail. For now, we're just making observations. The next one will be potassium. We can see that also cultivation and herbicides are affecting your potassium distribution. The till versus the no-till doesn't seem to have an impact on potassium. In the case of calcium, we're seeing that tilling does have some impacts on your calcium present in your soil, and then we're also seeing cultivation and herbicide affecting your calcium distribution. We're gonna look at magnesium next, and we're also seeing some significant differences. When we just mow under the vine, we tend to have much more magnesium available versus when you cultivate or use herbicides. And similarly, tilling seems to have, in this case, a little bit of a benefit, but it doesn't seem to be significant. So we will say that magnesium is not affected until based on this study. And then more importantly, we want to look not only at these individual nutrients, but we're also want to be looking at the ratio of them. Calcium, magnesium is one of those important ratios where you want to have, similarly what Chip was explaining about the nitrogen and the carbon, if you have too much magnesium, then you start having calcium deficiencies. So usually we want to have about 4 to 5 times more calcium than magnesium. And here we can see that tilling affects that ratio. We can see that tilling would decrease that calcium magnesium ratio. So in this case, you will start seeing some calcium deficiencies. And what this is going to cause is that eventually you're going to have to be inputting more calcium, versus if you don't till, you will see that you would have a little bit more of a steady presence of calcium and magnesium. In the case of cultivation versus herbicide, we actually see the opposite. Cultivation seems to increase calcium magnesium, which it's a little bit odd because cultivation is essentially tilling. Like I said, very preliminary results. We're still working on it. We're still collecting more soil samples. So we don't want to look too much into it, but we're seeing that in this case, cultivation seems to have a greater calcium magnesium ratio. The next one would be potentially mineralizable nitrogen. Nitrogen, it's not that important in vineyard systems, but it becomes a little bit more important on other systems. Nevertheless, it's an important parameter to look at because the amount of nitrogen that is potentially mineralizable in your soil, it's an indication of your soil microbiological activity. So even though nitrogen is not that critical in vineyard systems, knowing how much of that nitrogen can be mineralizable by the microbes will give us an indication of how healthy your soil is. So in this case, we're seeing that no-till has a significant difference when compared to tilling. So if you till your soils, you are going to significantly decrease your potential mineral nitrogen. And this makes sense because you are essentially disrupting the microbe balance that was created in the soil. So every time you rotate your soil or your soil gets disturbed, your microbes that were in equilibrium with a certain amount of oxygen get exposed to more oxygen, things get out of whack. So that's when you break that balance. And we're seeing that cultivation has a less impact on potential minerals of nitrogen than herbicides. And something that this could be explained by is the fact that when you apply your herbicides, you're essentially disseminating all the activity in that region. There's no living roots. Your organic matter might be lower as well. So you are not allowing for these microbes that will process the nitrogen, those nitrogen fixation bacteria, to do the job properly. So that's something to look at as well. I wish I would have just mowing as a comparison, but we don't have it yet. We'll be able to incorporate to see if mowing would have a less impact on microbes than cultivation of herbicide. The next one would be cation exchange capacity. As Chip mentioned, it is very important for the soil to be able to retain and be able to exchange nutrients within the soil in order to allow access to the vines. So if you don't have sufficient cation exchange capacity, you will not be able to have storage of nutrients and the necessary mobility to keep your vines happy. So in this case, we're seeing that not tilling would have better cation exchange capacity than tilling. This makes sense because every time you disturb the soil, you are removing by default some of that organic matter that would allow you to have sites where the cations can stay and mobilize around. And then we can see that herbicides would have a less impact on cation exchange capacity than cultivation. Again, eventually we're going to have a comparison with mowing just to see what having those living root systems in the soil will do for cation exchange capacity. Okay, we're gonna move on to total organic carbon. Again, in my opinion, this is one of the most important parameters because all physical, biological, and chemical properties are gonna revolve around carbon. So if you can maintain proper amounts of carbon in your soil, then everything else will sort of fall into place. And ways by which we usually recommend to keep carbon in the soil, it's by reducing tillage. we're seeing it in this little study that just eliminating tillage tends to increase almost 0.5% of organic matter. Now, 0.5% may not seem a lot to a lot of people, but as Evelyn mentioned, just increasing 1% organic matter in the soil would allow you to increase your water holding capacity by 25,000 gallons per acre. So 1% definitely goes a very long way. And when it comes to increasing your physical, biological, and chemical properties, 0.5%, it is quite significant. So we're seeing that tilling is definitely affecting how much carbon we have in the soil. No surprises there. We knew this all along. And then we're seeing a steady increase in the effect from cultivation to mowing. So cultivation versus mowing tend to have about also almost 1%. And then between herbicide application and cultivation, we're seeing also some impact there. So essentially what this is showing us is that when it comes to carbon in your soil, tilling, it's not helping. And cultivation and herbicide applications are also not helping. Leaving a living cover crop under your vine that is just mowed allows you to keep more of that organic matter. And that makes sense because if you're not disturbing your soil, then you're not releasing it. If you're not disturbing your soil, you're not releasing carbon into the atmosphere. But also by leaving these living roots in the soil, you are exuding more carbon, and that is helping you maintain a good steady amount of carbon. Some of the preliminary results that we have say that about the average for Napa County is about 2%. So we're looking at the average for the entire region. It's about, well, that's for not only for Napa, but for Mendocino and Sonoma as well. It's about 2% organic carbon. And that is what the experts will say it's a healthy level of carbon. The next one we look at is going back to what Chip was mentioning is the carbon to nitrogen ratio. This is very important because when your carbon to nitrogen ratio falls below maybe 11 or 10, then you start seeing a little bit less biological activity towards carbon processing, and then you might start seeing some deficiencies. So we can see that tilling significantly changes your carbon to nitrogen ratio. This is only going from 12 to 11, but it's a significant change. And for some strange reason, we saw that mowing was affecting that. So this is something that we need to look into a little bit more and figure out the logistics of it. And as we digest the data a little bit more and we incorporate more soil samples into it, we'll be able to see it a little bit better. But for right now, we're seeing that cultivation herbicide doesn't seem to affect your carbon to nitrogen ratio, but tilling definitely does. So if you are having trouble maintaining proper carbon to nitrogen ratios in your soil and you are tilling heavily, that would be the first alternative to make changes. The next one would be active organic carbon. And this is essentially the carbon that is gonna feed the microbes. The total carbon is carbon that feeds the microbes, but also carbon that is more stable in the soil, that it's doing more of a physical effect, leaving sites for the microbes to live, etc. The active organic carbon, it's actually feeding the microbes. So it's the carbon that it's fresh, is new, has just been added. And this is very important for, it's an energy source for the microbes. So it's something that we like to look at. In this case, we're seeing that, again, tilling has an impact on the amount of active carbon, so does cultivation and herbicide. So if you want to keep proper amounts of carbon, active carbon in your soil, tillage reduction, and then incorporating some sort of permanent cover crop under the vine or reducing the amount of disturbance or herbicide application will benefit your active carbon. The next one that we're going to be looking at is soil respiration. And soil respiration, it's important because it gives us an indication of your biological activity. When microbes are actively working in the soil, they're going to respirate carbon dioxide, and that's what we measure in the soil. So the amount of soil respiration indicates the amount of biological activity in the soil, which in turn relates to how healthy your soil is. The more respiration, the more biological activity, the healthier your soil is. So we're seeing that tilling your soil essentially cuts down your soil respiration by half. And there's no surprises there, because when you disturb your soil, you're affecting that balance, that delicate balance that was created over time in your soil. And that will in turn affect how happy your microbes are in the soil. And then we're seeing also that cultivation, just like tilling, will have a greater impact. Herbicide doesn't seem to be affecting as much. This is important to look at, because every time, whether we like to hear this or not, every time you till, you are disrupting your soil. You are affecting the biological balance. You are releasing carbon. So every time you disturb your soil, you're going to see some impacts on all of these physical, biological, and chemical parameters in your soil. Now, we're not here to tell you to stop tilling. We're here to provide the information for you so that you can make more informed decisions, because there are benefits of tilling and there's reasons why growers do it. What we're trying to do is give you the information so that you can decide when is the best time to till, if it's worth it, understanding that every time you till, you're going to have some impacts. Now, not all tillage is done the same, depending on how often you do it, how aggressively, the level of moisture in the soil when you till. These things might shift around. What we're seeing in consensus is that tilling does negatively impacts your soil. So if you must till, keep in mind that you are impacting your general soil health, so that you can either minimize it or try to selectively decide when is the best time to till in order to get the best benefit with the least impact on your soil. Another thing that we're looking into that we don't have yet results on that is we're looking at the length of tilling. If you have been tilling for 10 years versus if you're tilling for 1 year, we suspect that there's going to be differences. So that's another thing that we are studying. Right now, this data only shows till, but all of this data can show 1 year or can show 10 years. So we eventually are going to try to disseminate that and see where the differences are if you till for 1 year versus if you till for 10 years, what the impact is. If you are in a no-till system, then what happens when you start tilling again? Are you going to lose all of your organic matter? Is your soil microbiological activity going to be affected right away? Is it going to take time? We're going to be able to answer some of these questions as the project progresses. So some of the conclusions from this study that we have done so far is that soil disruption negatively impacts soil health. And this is going to affect your water aggregate stability, your cation exchange capacity, your soil respiration, your carbon, and some nutrients, particularly nitrogen. So however you see it, every time you disturb your soil, whether it's with tilling or cultivation on the vine, you are affecting your soil health in particularly these parameters. And we are seeing that maintaining a permanent cover crop on the vine has the potential to benefit soil health. So keeping those living roots under the vine are the best thing that you can do for yourself. Now, logistically speaking, I understand that it's very complicated in some cases to maintain a permanent cover crop. You need certain specific equipment. Sometimes you have to import it from other countries in order to have that mode. Again, I'm not here to tell you what to do. I'm just here showing you this information and showing you that keeping those living roots in your under your vines have the greatest benefit for your soil. It is up to you to decide how this will get done and if it's possible for you. Some of the things that we want to do in the future will be, we're collecting additional soil samples. This year, unfortunately, given the issue with the quarantine, the lab that was analyzing the soil samples had to shut down, so we had to stall on the soil sampling. But we hope that next spring, we're gonna be able to collect another 100, 150 samples. Those samples will be added into the database, and we're gonna keep refining it and figuring out the true impacts of these management practices on soil health. At the same time, one of the things that we need to do is to separate this data by soil type and region, because not all soil types have the same capacities. You're going to have some soil types that are more capable of storing carbon, some soil types that are more capable of being able to increase water holding capacity, nutrient holding, etc. All of these parameters that we described, they are very, very much linked to the soil type. So everything that I show you shows every single soil type that we have sampled. So we need to disseminate the data so that we separate them by soil type, so that we can start seeing that if the impacts that we're seeing are just because of the management practices, or is it because the soil type differences? So if we go into a rocky soil, that's going to have very much different capacity to hold carbon and manage the other parameters versus a clay soil. And then if you go in between, then you're going to have different parameters. So in the near future, hopefully by the time that we do this workshop next year, we'll be able to provide you with a little bit more information on how the different soil types affect these soil health parameters. And when we develop the database will incorporate that, so that if you want to utilize the database, you would only have to look for samples that sort of match your same soil type, and then you can compare yourself to that. Because if you compare yourself to other soil types, that's like comparing apples with bananas. There's not a direct correlation with that. So a lot to come. This is a project that has been going on for almost 5 years now, and we still have a couple more years to go. So please stay tuned on that. Another important aspect of this project is that we have demonstration sites in order to test the impacts over time of these management practices. Here in Napa, we have 2 demonstration sites, Gamble Ranch and our own vineyard in Carneros with Chica Creek Vineyard. And we're demonstrating different things. For example, at Gamble Ranch, they were under a no-till system. And we asked them to give us a small section of the vineyard and start tilling it. So in that case, we're gonna assess how going from a no-till system over, I think, 10 years or so, looks like once you go into tilling. So that way, we're gonna get a direct indication of how much tilling actually impacts the soil. Is it a direct impact? Is it an impact that builds over time? That's the question that we're gonna be addressing with that. And then in our own vineyard in Chica Creek, we're looking at several things from the effects of compost and or biochar applications. We also have a till and a no-till system. We have rows that have not been tilled since 1995, right next to rows that are tilled every year. So in that case, we have a direct indication, side to side, same block, same vineyard, how tilling it's impacting the soil health, but also how this is happening over time. And there's other trial sites going around in the other counties. If you are interested on participating on some of these trials, or you want to learn more about these trials, please let me know. We're always looking for interesting vineyard managers that would like to experiment in their vineyard. And like I said on all my other workshops, we try to provide information for you, but if we do make some recommendations or we suggest you do something, feel free to try it in a small scale. You don't have to go for your entire vineyard. You can try it on a couple of rows, see how it works for you, because there's so much variability in Napa County that it's hard to pinpoint just one thing that will work for everybody. Your operations are different, you're trying different things. So we want to make sure that we let you know that it's not everything we said is going to apply to everybody, but if you try it in a smaller scale, then you can use that to convince yourself if it's going to work for you or not. So with that, the only thing that I can tell you is that based on our study, the best way to keep your soil happy and healthy is to keep it covered and to keep it undisturbed. And this is going along with what the NRCS is suggesting as well. If you can look at this picture, I cannot see the soil. I cannot see it. It's either covered or it's not being disturbed. And this is something that my old college professor would say to me is, any soil that you can see, it's a vulnerable soil. If you can see your soil is exposed to the elements, and that soil will eventually be affected. And there's nothing we can do about it. So if you cannot do a cover crop under your vine, try mulching. Try other methods by which you can keep your soil covered. And if you're looking at the middles, if you have a permanent cover crop, if you just mow it, that's the best thing that you can do for your soil when it comes to soil health. The practicality of doing some things and how things are done, that we leave it up to you to see what makes more sense for you. But I want you to walk around your vineyard, and I know you always do it, but I want you to walk around your vineyard. And I know that we usually say, look up, don't look down, but I want you to walk your vineyard, and for that entire time that you're walking your vineyard, just look down, look at your soil. And if you can see your soil, think about how all of these principles that we taught you today are affecting the health of your soil. And think about how you can change that. Think how you can cover that piece of soil so that it's not being vulnerable. And above everything, try to keep it undisturbed as much as you can. And that is the best thing that we can offer to you as far as what you can do with your soil. So with that, I thank you for your attention, and we're gonna open it up for questions right now. Let me go ahead and stop sharing this. So that is the end of the presentations that we have for you today. We still have about an hour to go. We don't have to do it for a whole hour, but I'm gonna start addressing some of the questions that you have. Feel free to ask questions for me or for Chip and Evelyn, and that way we can get the conversation going. So let me look here. Let me go all the way up because there's quite a few questions here. Okay, so here's the first question. When you talk about 10:1 or 20:1 ratio of carbon to nitrogen being ideal, what specific soil lab analysis will you use to assess that ratio? That's a good question. We have a lot of commercial labs. Luckily here in Napa, we are sending our soil samples to Oregon State University because this is more like a research project than anything. So we wanted to make sure that we have a researcher doing these analysis. But there's a couple labs. You have Delaval, you have A&L labs. There's quite a few around the area. And when you are looking at this, you can ask them specifically to test for you the carbon to nitrogen ratio. But what they're gonna do, they're gonna measure the carbon and then the nitrogen separately. And there's different methods by which they do that. The most common one is due by combustion. So they'll take a small sample of the soil, they wrap it in aluminum foil, and they burn it to the ground. And then they weigh it, and then they measure how much of the nitrogen and how much of the carbon got lost through that. But if you send your samples to a local lab, you can ask them to do that for you. Let's see. What are you measuring in the samples? So I think I went through that. This might have been an early question, but when we're doing these samples, we're measuring all of the different parameters that I listed. We're measuring all the nutrients, NPK, calcium, magnesium. We're measuring the calcium magnesium ratio. We're also measuring water holding capacity, aggregate stability, biological activity based on soil respiration. So that list that I gave you is essentially all the samples that we are measuring. And you're more than welcome to send them to Oregon State University. We pay $150 per sample because it's much more thorough. It has way more than a grower may need. I don't necessarily suggest you do that. All the local labs do a really good job. It's just that they might not be able to do certain tests that we do, especially, for example, the respiration. It's something that not many labs do around here. They sell kits that you can do it by yourself. If you're interested, we can talk more about it. Okay. Would you be looking at herbicide, fungicide, and other chemical use in the sampling program? So right now, the way that we're breaking it down is herbicides, and we're not discriminating between organic and non-organic. If it's an herbicide, we categorize it as such. And then we're looking at cultivation and just mowing under the vine. Those are the only parameters that we're looking at. Fungicides, they tend to be applied foliarly. Some of them might make it into the soil. That's a good question. Maybe for a different project, we might be able to do that. So Carmen is asking about Marty's question about in fertilizer and the impact of in fungi. Carmen, we did address that. I encourage you to go back to the video, and if you cannot find it, you can email me and I can talk to you about it. Again, somebody's asking about fungicides. We are not doing it at the moment, but it's something to consider for future projects. And somebody is mentioning about a particular study. I don't have time to address that right now. Pam, please email me and I'll be more than happy to look at that study and then I can give you my opinion. So what does mowing represent? So the way that we're looking at it for the undervine is if you cultivate, meaning you bring some sort of disruption into the soil to cut the weeds that are growing, either you do that, you do herbicides, or you do nothing, and then you just mow on top of the crop, maybe leaving it a couple inches tall, so that you're leaving all the living roots intact under the vine. Now that is, I understand that's a little bit tricky. You need specialized equipment to do that right under the vine. Again, we did find some growers that just mow under the vine, meaning they just keep it a couple inches high and that's it. They don't cut it any deeper. They don't incorporate it. They don't disturb the soil. So mowing in this case essentially represents not touching the soil at all. You leave it intact, you leave it undisturbed. So that represents the best case scenario when it comes to maintaining your soil health. Are they separate breakouts for organic growers? Not at the moment. So when we're looking at these, like I said, we're looking at just those 5 parameters, till versus no-till, cultivation, herbicide, or mowing under the vine. So within those categories, somebody that does not apply herbicides and decides to mow, most likely is organic. But other than that, we are not distinguishing between organic and inorganic growers for the moment being. We do have that data, so we could break it down at some point. We're also looking at compost and other amendments applications, which a lot of organic growers do. But because we're not looking at the direct impact of, like I said, herbicides, we're just looking at them in general. We don't care if it's an organic or an inorganic herbicide. So in that case, we're making a little more generalized, and we have to keep it a little bit generalized in order to keep the conversation going. But as we progress on the project, we might get some ideas of what other parameters to look at. We do have the data, so it will be easy to do. I've been asked, which method did you use to measure the soil phosphorus? So we send it to the lab, and they use 2 methods. I don't have it on top of my head, but if you email me, Lucas, I'll be more than happy to tell you. Because I don't really do it myself. I just collect the samples and send them to the lab. I don't really worry much about it, but if you email me, I can tell you specifically which methods we're using. I can share with anybody that is interested. We have a long list of which analysis we're sending to the lab to be done and how they're doing it, because that is important. That's an important distinction. If you want to compare your lab results, it has to be done in a similar way. So I can definitely share that with anybody that is interested. Just send me an email and I'll be more than happy to help you. So what is the difference between tilling and cultivation in your grass? So for all intents and purposes, cultivation and tilling is essentially the same. It's a way to disturb the soil to eliminate weeds or cover crop and incorporate it into the soil. I just like making the distinction because tilling tends to be a little bit more aggressive. When you do cultivation under the vine, you tend to just do enough disruption of the soil to remove whatever weeds you have. Sometimes if you go with weed whackers, you don't really disturb the soil. So we consider weed whacking mowing essentially, but for the intents of this project, I just did it as a distinction, but in the end, it's just disruption of the soil. Next question, where do fungicides figure in this? That's a good question. I guess it's becoming more and more popular. We are not considering that, but I'll keep it in mind for future projects. And I get another study. So again, I don't have time to address this study right now, but I will read it. And then if anybody's interested in my thoughts on that, feel free to send me an email and we can talk about it. Somebody's saying, aren't herbicides disturbing the soil? If you use herbicide, the fungi are hugely impacted. Yes, so we definitely think and we are observing that there's a lot of soil health indicators that are being affected by the use of herbicides, just because when you use herbicides, the first big impact is that you're eliminating the chance for any living roots to be in the soil. So when you do that, you don't have enough organic matter being distributed, exuded by the roots. Your microbes are not happy, but I also suspect that some microbes may be impacted. Don't quote me on that though, because I don't have enough information to go on that, but I suspect that the herbicides are affecting just the general microbiological activity in the soil. And we're seeing some of that in our study. Somebody's asking, why does herbicide application not affect the soil respiration and also the active carbon and the soil organisms helping the respiration and carbon levels? Really good point. I was a little bit perplexed by that result as well. But right now, we're still collecting more soil samples. We, like I said, we're gonna break the data into different soil texture types and potentially by region as well. So that way we'll be able to get a very better idea, a clear idea on how this is specifically impacting. So for right now, Carol, I don't really have an answer for you. I do agree with your hypothesis. We just need more data to figure out what's going on there. We also need to clean out the data a little more. If you notice on the data, there's no margin of error. I need to figure out what the margin of error is. If there's any outliers in the data that are kind of like skewing the data somewhere or another. But in general terms, yes, we should see that herbicides will affect the soil respiration in the active carbon internal lab. So more to come. Stephen is saying tillage shreds the arbuscular mycorrhizal fungi networks. Yeah, I mean, every time you till, you're disrupting your soil structure and building carbon in the soil and maintaining a proper balance of microbes and all the nutrients. It takes a long time. So when you come in and disturb your soil, you're essentially undoing all the hard work that your soil did over that amount of time. Again, I'm not shaming anybody for tilling. We're just providing the information and trying to help you make an informed decision. The bottom line is that every time you disturb your soil, you're going to have some sort of negative impact on your soil health. That's just what we're observing in general terms. Let's see. When you're measuring tilling, does that mean in the rows between the vines or the strip that is under the vine? So when in this study, tilling is specifically for the meadows. Under the vine, we are calling it cultivation. Are farmers leaving cover crops directly in the strip under the vines? Some of them are. So for those that I identify as mowing, what they do is they keep a permanent cover crop under the vine, and then when the cover crop is getting too high, they will go ahead and just mow it and then leave a couple inches of the ground so that they're not disturbing the soil and leaving the roots intact in there. What about growers tilling every other row? That's a really good point. So for our trial demonstration site in our vineyard in Carneros, we do have alternate tilling. However, the rows that are not tilled never get tilled, and the rows that get tilled every year get tilled. Some people would do rotation. So every year or every other year, they will alternate. So in that case, some rows get tilled and then the other one doesn't get tilled, and then they will switch every so often. That is a very important distinction because the length of time in between no till will definitely allow the soil to develop more of these organic structures and increase your aggregate stability, all of these things that we talk about. So the question is, when you come back, if let's say I don't till for 5 years and then I come and till, do I lose all the benefits? Is it a progressive thing? We're trying to study that. And in our data, like I said, for the till rows or the till graph, that includes everybody, whether you till every year and you rotate or whether you rotate every 3 to 5 years. So that's another piece of information that we need to disseminate. We're gonna break it down into the different years in between rotation of the tillage. And that is gonna give us a good indication of the length of impact based on the length of the practice. So that's a really good question. More to come on that. Is the soil dramatically different between those rows that are tilled and those that have permanent cover crops? So the soil itself is the same. It's the same texture between your rows, your undervine, and your meadows. However, we're seeing significant differences on the soil health in those two sites. And that's because they get managed differently. If you manage them differently, there's gonna be different impacts. So the soil itself, the texture, is the identity of the soil is the same. It's the biological and the chemical parameters that got affected that in turn affect the physical properties. So even though it's the same soil, they have different quote unquote scores of soil health, and therefore they're going to perform differently. Let's see here. Do most growers use permanent cover crops or annual cover crops that are replanted every year? That varies a little bit here in Napa. There's a big movement to use cover crops during the winter. So almost everybody uses a seeded cover crop, at least, but for those that are under no-till, they'll have more of a permanent cover crop in their systems, but it really varies. The good things is that during the winter, most people keep it covered. Especially if you're in a hillside scenario, you have to by regulation, but for those in the valley floor, they do it by choice because they understand the benefits. The issue comes in, how do they determine in the cover crop? Do they come and just mow it if it's a permanent cover crop, or they will come and till it if it's not a permanent cover crop? That's what the differences that we're seeing. Somebody's asking me, can you share DOE and ANOVAs for the test variables? I'm not sure what is that, Jim. Can you explain to me what those acronyms mean? I don't know what that means. Why not measuring fungicides? Thank you. We will definitely come back and think about how we can incorporate fungicides. So far, we haven't done it, but we intend to keep these projects going for many years to come. So the Soil Health Hub will keep going and the work will keep going. So I will make sure to make a note of that. It says a lot of people stop herbicides but continue fungicides. That's the thing. I mean, when it comes to herbicides, there are alternatives. You have options. When it comes to fungicides, it's a little bit difficult. Even if you have an integrated pest management plan, fungus, it's going to be one of those things that are definitely going to be affecting your vineyard. So it's a little more tricky to get rid of fungicides, but we'll keep it in mind. Let's see here. Oko is mentioning, do you incorporate, forgive my pronunciation, huge culture method or Ruth Stout method of no-till heavy mulch gardening? Do you advise against bonamine as fertilizer due to the mad cow disease implication? So I am not sure about any of those things you mentioned, Oko. I'm sorry, I'm just not familiar with them. In general terms, I would incentivize mulching under the vine or in other areas of your vineyard if possible, just to keep your soil covered. So the ultimate goal is to keep your soil covered. And if it's covered with living roots, it's even better. But if you cannot have that, just having it covered with the mulching, wood chips, or any other thing that works for you. Straw mulching, it's not recommended because it doesn't last long. You can use it for winter, so like to be ready for the rains, but not under the vine as much because you will disintegrate faster. It has a lot of nitrogen, so it would affect your nitrogen balance. Wood chips are a really good way of doing it. Big 1 inch to 2 inch pieces that will not disintegrate. Tomorrow, we're going to have Will Bax from Marine Compost talking about the benefits of mulching and composting. So stay tuned for that. And I'm not sure about the bonamine and the mad cow disease. I'm sorry about that. Pam leaves me a note. It says, Miguel, no-till often means herbicide. What do you recommend? Well, not necessarily. If you're under no-till, you can mow. You can do hand weeding, which again, I recognize is not 100% practical. Mulching is another way if you are not under no-till. But we have to make a distinction between the no-till in the middles and the no-till under the vine, because those are two very distinct management approaches for vineyards anyways. So if you're in the middles and you're under no-till, then if you establish a permanent cover crop and then you just mow it, that's the way to go. It might take a little bit of time if you go from a till to a no-till operation to establish a permanent cover crop, but once it is established, you shouldn't have any problems with it. Under the vine, it's a little bit more tricky because the cover crops, you need to make sure that you are not hosting any pests in those cover crops. So I recommend you reach out to your local cooperative extension. Here in Napa, we have John Roncaroni who focuses on weeds. Unfortunately, he's retiring soon, so let's try to get some of his knowledge before he goes out. There's some species that we are experimenting with in our vineyard. The soil fescue is one of those that may not be aesthetically pleasing, but it doesn't grow much under the vine, so you can keep it down. We're also experimenting with these other grass species that goes dormant when the vines wake up. So you can put it, it's a very short grass. It's a Pua bulbosa species. We're experimenting with that so that we can keep sort of like a permanent cover crop. Now in our vineyard, we stopped using herbicides a few years ago, Roundup specifically. We are experimenting with weed slayer, which is an organic herbicide, but it's really, really difficult when you have high weed pressure to incorporate a cover crop. It's just gonna outcompete, the weeds are gonna outcompete the cover crop. So it takes time. And again, I understand that logistically, this may not make perfect sense. What I hope with this is that you can understand the benefits of at least exploring some of these ideas. And I'm a firm believer that if I can show that these concepts are true and that they work, somebody will come up with a way of doing it. For example, we have an experiment in our vineyard where we are using wood mulch, wood chips as mulching for weed prevention and some moisture retention as well. We have a lot of weeds, so we're using that. And I did it in 2 acres. So that we apply it by hand. And it was a little bit expensive, but not too expensive. Now, if you're gonna do it in 100 acres, that's gonna be a little bit more complicated. So the practicality of it is not something we're discussing of today. I'm just discussing the benefits, and hopefully somebody will come up with a good way of doing it. And that way we can get the ball rolling. But the bottom line is that when you keep your undervine soil uncovered and it's completely bare, you are affecting the soil health and you're affecting the ability of your soil to do its job. Now, I have been here in Napa County for 1.5 years now. Before here, I was working for the RCDs in the Coachella Valley in the Inland Empire, which is a more dry environment. And we do have table grapes and a little bit of wine grapes in Temecula, but not a lot. But I work with a lot of different crops over the years. I've been doing this for 5 years now. And one of the things that I noticed when I came here to Napa for the first time is that the undervine soil gets completely neglected. And that's just my personal and professional opinion. If you drive around, you're gonna see that the undervine, it's always for the majority of the vineyards anyways, it's kept completely bare. And one of the things that I started speculating is that most likely that soil, it's not, that's what I thought when I first came here. Most likely that soil under the vine, it's less healthy than the soil right next to it in the middle, because the middles are kept, even though you might till it, in my opinion, the most important piece of soil is right under the vine. Because when you irrigate, you are doing drip irrigation is right under it. When you apply nutrients, you might do it through fertigation, and that goes through the drip as well. So the majority of folks, unfortunately, in my opinion, are neglecting the most important piece of soil, which is under the vine. Now, that is by design, I think, because it's difficult to not, because the easiest way to do it is just go and apply herbicides everywhere under the vine, forget about it, and then you move on or cultivate. And what we're trying to see with this study is that it is actually impacting significantly. We're seeing that the undervine, in general terms, the undervine soil is considerably less healthy than the middles. Regardless of what you do with the middles, that's just what's happening. So I really encourage everybody to keep a closer look under the vine. The soil under the vine, it's the most important soil, in my opinion, that you have in your vineyard, and it's the soil that should deserve the most attention from you. And I understand that it's a lot of work to keep either covered or a cover crop under the vine, but I hope that we can incentivize you to give it a try. And like anything else that we suggest, you don't have to go and do it in your entire vineyard. You can try it in a few sections. Our vineyard in Carneros, which is a creek vineyard, we use it as a demonstration vineyard. So if anybody wants to come and take a look at these crazy things that we're doing, or if you have a crazy idea, let us know, and we are more than happy to explore the possibility of trying it in our vineyard just for the sake of demonstration. We understand that there's a lot of risk involved. You guys run businesses. This is not, you're not doing it just for fun. So we want to make sure that your economic integrity is not impacted as well. but we do want to incentivize you to keep a good look at the soil under your vine because it's being impacted. I have another question here. Can you say a bit more about the trade-off between weeds and cover crops? Can the cover crop become a weed, or can the weed technically become a cover crop? I love that question. That's amazing. If you look at the definition of weed, you're gonna find hundreds of different definitions. So technically a weed is an undesirable plant species in an undesirable spot. All plants have native habitats. If you find a weed in its native habitat, it's not a weed. It's functioning properly. It's doing what it's supposed to. Unfortunately, agriculture, we get exposed to natural environments, and then we get plant species that are native and some of them that are not native. But because they're undesirable in our vineyard, for whatever reason, because they host pests, because they compete with the nutrients and water in our vines. But the bottom line is that having some living roots in the soil, it's much, much better than not having any. So if your soil is completely covered with weeds, I am pretty confident to say that that might be a healthier soil than if you are spraying herbicides to disseminate everything. So in that concept, yes, weeds can be treated as cover crops, and cover crops can eventually become weeds if they get out of control. The problem with weeds is that they are, by definition, resistant to methods that we like to use in order to get rid of them. Some of them might be resistant to weeds, or they're just very, very strong. So in that sense, they could bring problems because if they grow too much, they can affect the vines. But also if they're hosting pests, then you don't want to have that. But there's cover crops that may also hover pests. So it's important to educate ourselves about the type of cover crops that we want to plant in our vineyards, and if there are any issues with potential pests. Having said that, my way of looking at it is it's better to have it covered with something. And if you go to our vineyard, you'll see an example of how weeds can just take over everything. And we're working really hard to keep them down. So for the most part, we just weed whack them and try to keep them down. But oh boy, they come back all the time. But if you keep leaving roots, your soil doesn't know it's a weed. You know, your soil is just receiving the benefits from whatever plant it's living above it. And these plants are gonna exude organic material as well. And these weeds, unfortunately, are gonna be competing for nutrients and water as well. Different species may do so more strongly than others. But in just the general terms of soil health, it's better to have weeds than have nothing. When it comes to the practicality and other impacts, I'll leave that up to you, but it's better to have something. If you don't want to deal with weeds, the next step would be to use wood mulching or some sort of other mulching that will keep your soil covered, but you don't have to maintain it. You don't have to worry about any potential pests or anything else. Although, as we're going to learn tomorrow, there's some things to consider also when you're applying mulching. It's not all good. But like that picture that I showed you, if you walk around your vineyard and you see pieces of soil that are exposed, think about how would be the best practical way for you to cover that soil. How can you protect that soil from degradation, and how can you do it in the best possible way that it's going to be economically and just physically possible to do it. There's no one single answer for any of these questions. It comes from a case-to-case basis as well, especially if you have heavy weed pressure. If you want to just apply mulching, it's going to be a learning curve, and you're going to have years where you're going to have to maybe incorporate a little bit of herbicides here and there, or mechanical weeding here and there. A lot of these things, any transition period will definitely be challenging, going from one extreme to the other one. But like I said, if you try it on small portions of your vineyard, you can learn what is working, what is not working, without risking the integrity of your operation. That's my best advice that I can give you. Just try it on a small scale and see if it's working for you, or if it's not working for you, and then move on. And some of these things that I said, as far as suggestions on how to improve soil health, may not be practical for you, and that's absolutely fine. My objective with this is so that you can have the knowledge, so that you can at least think about it and maybe stop for a second before you go and till, or stop for a second before you decide to apply herbicides and decide, okay, maybe I don't have to till today. Maybe I can wait a couple months, or maybe I don't have to apply herbicide today. And any reduction on tillage and any reduction on herbicide applications will have a benefit. So you don't have to go to the extreme and eliminate tillage or eliminate herbicides. If you cut it by half, that's a huge benefit for your soil. More importantly than anything, I think that everybody needs to assess their soil health more often. And just like you send pedio samples every year, I suggest you send soil samples every year as well. And there's certain things that we can do for you. We can assess certain things, water infiltration, compaction, those things we can assist you with. And we can either help you collect soil samples, or we can help train your team, your staff, how to collect these soil samples. We can offer all of that at no cost to you. In the past, for those that participated in the study that I mentioned, they received the soil lab analysis for free. And I wish I would have the money to offer that to everybody. We're working really hard to get more funding so that we can in the future offer at least basic lab analysis for free. Unfortunately, we don't have that right now, but we do have a lot of local labs that are relatively affordable. So if you don't know really approach making changes in your vineyard as far as soil health goes, the first step would be to collect soil samples, send them to a lab, work with us, work with the NRCS, work with your cooperative extension, so that you can understand where you stand. And that way you can make more informed decisions. If you don't know what's going on in your soil and you're just speculating, then your solution can only go that far. So the first step would be to collect soil samples. Like I said, we can offer that at no cost as far as guiding you through the process. You would just be responsible for the lab fees. But in the future, hopefully we can have funding to do that. I don't have any more questions here. I'm gonna give you guys 5 more minutes to see if you have any questions, comments, concerns, and then we'll end the workshop. We're trying really hard to bring you information, latest scientific information for these workshops. We will be sending you a questionnaire with just asking you your experience with this workshop and recommendations. So we will be very, very grateful if you can complete that workshop and just give us recommendations of what you would like to see on future workshops between the RCD staff and the NRCS staff. We have a lot of knowledge and we can cover pretty much all topics that you may be concerned about. And if we don't have an answer, we work really closely with the UC Cooperative Extension, with the Ag Commissioner, with other agencies where we can get answers for you. But please let us know what else you would like to see. And remember that this is only day 1. Tomorrow, we are gonna have a couple other presenters. We're gonna have Bill Birmingham from the RCD talk about soil erosion and soil prevention. We're gonna have Kelsey Brewer from UC Davis talk about the benefits of grazing sheep in the vineyard. And we're gonna have also Will Bax from Marine Compost talking about the benefits of mulching and compost. We're gonna have Josiah Hunt from Biochar, Pacific Biochar, talking about the benefits of biochar. And then we're gonna have Jeff Creek from the Carbon Cycle Institute talking about carbon sequestration, carbon farm planning, and how increasing carbon in your soil can benefit your soil health. So very, very interesting group of people talking tomorrow. So I hope that I can see many, if not all of you guys tomorrow as well. Okay, so it looks like we don't have any more questions. I'm gonna go ahead and add my email address to the chat here. And also my phone number so that you guys can contact me if you have any questions. And I'm really good at responding to emails and phone calls. If I don't answer your phone call, just leave me a voicemail and I'll get back to you very shortly. So I'm gonna leave my contact information right here, and then we're just gonna leave it for a minute, but this concludes today's workshop. We'll continue tomorrow. Thank you so much everybody for your time. We really appreciate it, and we're looking forward for your input as well to see how well we did with this workshop and what else you would like to see. I hope you guys have a good day, and I hope I can see you tomorrow.